My name is Taylor Pack. My video presentation will be on Retroviridae. Retroviridae is the cause of the retrovirus. The retrovirus, like any virus, is an obligate intracellular parasite, which means it's dependent on the host for replication. Here we'll look at the structure of the retrovirus. It's a single-stranded positive sense RNA virus. On the outside is the envelope, which is made up of glycoproteins. These glycoproteins are embedded into this lipid membrane bilayer. The circular structure is the core shell protein, and inside of that is the core protein, and the string-like structures within the core protein are RNA, and embedded into the RNA are enzymes reverse transcriptase, integrase, and protease, and these all play a role in the multiplication of the virus. So the glycoproteins I talked about in the last slide are part of the first step of replication. These glycoproteins bind to the receptor proteins here in step 1. In step 2, once these have been bound, the cell membrane degrades and becomes part of the host cell. The RNA strand and the enzymes will then enter the cell. Within the cell, reverse transcriptase creates a complementary strand of DNA from the retrovirus RNA, and the RNA is degraded. This strand of DNA is known as cDNA. So here in step 4, the cDNA is then replicated, and the two strands will enter the nucleus. In step 5, once in the nucleus, the DNA is integrated into the host cell's DNA with the help of the enzyme integrase I talked about in the last slide. So step 6, the cell can either stay dormant, or the RNA may be synthesized from the DNA and used to create the proteins for a new retrovirus. In step 7, ribosome units are used to transcribe the mRNA of the virus into the amino acid sequence, which make proteins, and this happens in the endoplasmic reticulum. This step can also make viral enzymes and capsid proteins. In step 8, viral RNA will be made in the nucleus, and these pieces that I talked about in step 7, the viral enzymes and capsid proteins, are gathered and pinched off of the cell into a new retrovirus. It enters the cell here. It exits the cell here in step 9. And so when retroviruses have integrated their genome into their germline, their genome is passed to the following generation, which is when transcription is thought to occur from DNA to RNA, reverse transcriptase transcribes RNA to DNA, and so the term retro and retrovirus refers to the reversal, which is making DNA from RNA. So the different types of Retroviridae or retroviruses are HIV, which is human immunodeficiency virus, FIV, feline immunodeficiency virus, which affects cats, and BIV, bovine immunodeficiency virus, which affects cows. So these are just a few more well-known retroviruses, and there's numerous different kinds. These are just a few examples that affect humans and animal hosts. So transmission of the retrovirus is cell-to-cell transmission. So what is cell-to-cell? Transmission through bodily fluids that contain cells. So they're transmitted, here you see, by sexual contact with an infected person, through pass from an infected mother onto a child, or through breastfeeding, a mother who has it breastfeeding a child, and someone who has the virus that have used the needle, or and then the blood from that needle gets passed on to the next person. So these are all through bodily fluids. And cell-to-cell transmission of a retrovirus is much more efficient than extracellular infection, and because retroviruses spread through the tight cell-to-cell interface, they're out of reach of the immune system. Retroviruses are also being used as vectors for gene therapy. So up here in this, you can see at the top, retroviral vectors are created by removing the OP, the retroviral GAG, the POL, and the EMV genes. And these three genes, these are replaced by the therapeutic gene here. So in order to produce vector particles, a packaging cell down here is essential. Packaging cell lines provide all the viral proteins required for capsid production and the viral maturing of the vector. So these packaging cell lines have been made so that they contain the GAG, the POL, and the ENV genes in the retroviral genome. So following the insertion of the desired gene into the retroviral DNA and the maintenance of the proper packaging cell line, it's now a matter of preparing the retroviral vectors. But one of the problems of gene therapy using retroviruses is that the integrase enzyme can insert the genetic material of the virus in a position in the genome of the host that's arbitrary to what's being targeted. So if the genetic material happens to be inserted in the middle of the original genes of the host cell, this gene can be disrupted if the gene happens to be one of the regulating cell division, which causes uncontrollable cell division, and cancer can occur. So here I'll talk a little bit about how the cancer is produced. So the DNA from retroviruses can be incorporated into the host gene as the provirus that can be passed on to progeny cells. When the retrovirus DNA is inserted at random into the host genome, it can be inserted into oncogenes. So some retroviruses can convert normal cells into cancer cells, and some proviruses remains latent in the cell for a period of a long time, and it can be activated by the change in cell environment. And so tumor growth can occur. And treatment of retroviruses include nucleoside reverse transcriptase inhibitors, NRTIs, which are active inhibitors of reverse transcriptase found in the retroviruses such as the HIV, the human immunodeficiency virus. The different nucleoside reverse transcriptase inhibitors may be activated differently, but they all have the same mechanism of action. So reverse transcriptase inhibitors their mechanism of action is to prevent HIV from becoming integrated into the genome of the host cell. And this in turn keeps the cell from making copies of the virus and slows the progression of the infection. And there actually is growing problems with resistance to many drugs in these classes. Here I've named just a few of the NRTIs, the more well-known. They may not be very well-known because these are actually not the generic names.